This protocol demonstrates how to establish the iCRISPR platform for rapid genome editing in hPSCs and how to incorporate genome-edited lines with hPSC-directed pancreatic differentiation to study human pancreatic development and disease.
These methods can help answer key questions in the human pancreatic development field, such as the genetic control of normal pancreatic development and the molecular basis of diabetes.
The main advantage of these techniques is they allow us to efficiently modify genes of interest in hPSCs and to use these modified cell lines for studies of human pancreatic development and disease in vitro.
Culture hPSCs in chemically defined and feeder-free conditions on truncated human vitronectin-coated dishes. The day before electroporation, add 10 micromolar ROCK inhibitor during the media change. On the day of electroporation, prepare vitronectin-coated 10 cm dishes in advance. Then remove culture medium from the cells, wash once with PBS without calcium and magnesium, then treat the cells with 1x dissociation reagent at 37 degrees Celsius for approximately 3 minutes. Next, aspirate the dissociation reagent before the cells have detached and add 10.5 milliliters of complete medium. Use gentle pipetting to form a single-cell suspension before transferring to centrifuge tubes. Perform a cell count on 500 microliters of cell suspension using an automated cell counter.